So, welcome to the Ever Curious podcast. Today I'm with the other host of this show,
Oscar Reckel. Nice to meet you.
We're joined today by Dr. Anika Nagelkerke. She's an assistant professor at the University of Groningen, and she's working with biological models, and especially with organ on chip. Doctor, welcome.
Thank you.
So let's start with the classical question. Can you tell us something about yourself?
Something about myself, of course. That's an easy topic to start with, I would say. So my name is Anika, and I live in the northern part of Drenthe with my husband and my dog. He's 3 years old at the moment, so he's adorably cute and very big. I don't come from this region. I actually grew up in the province of Zealand, in the southwest of the country, in a very small village, not that many people. And I came to work at a university, which is something that I would have never expected would have happened.
Why exactly you wouldn't have expected that happened?
I'm a first generation student, or I was a first generation student, and now also, therefore a first generation academic, which in my family is not very common to do.
Wow, so you did not consider that before starting a university, that you would end up as a professor?
No, it was not a set out plan, definitely not.
And especially for an academic career, it's a very rigorous and hard career to follow, I'd imagine.
Yeah, for sure, yeah. So for me, it's also a bit, serendipitous, I would say. Yeah.
And what got you into the world of biological modeling, organ on a chip, and the research you're doing at the moment?
It actually happened during my master's that I did a research internship in a department, and it turned out that I really liked cell biology, and that was an area that I felt very comfortable in. And that just, it grew from there. I turned into a PhD, into a few postdocs, and now being an assistant professor.
And what was your PhD about, if I ask?
My PhD was more on therapy resistance mechanisms in cancer. So really, how do cells acquire resistance, and what are the consequences of that for their behavior, for their malignancy, more into metastasis, and seeing if we can hopefully help the world a little bit further on in this terrible disease.
Wow. So it's not quite related to the research you're doing at the moment?
It was sort of the start for the research that I'm doing now. So it laid the foundation for that.
Exactly, yeah. And that's also, me and Oscar always talk about that things are not set in stone. One can't change the direction of his research, and
for sure.
The disciplines are related to each other. So you could work in a multidisciplinary team, and you could change from, especially in pharmacy, we have chemistry, we have biology, and we have a lot of other elements also from physics. And it's interesting to us how we could specialize in one area, but we could also change directions and do other areas. So always talking about that list and hearing that actually is optimistic to us, I guess.
Exactly, and it also makes it exciting. You're not already immediately forced into a certain direction.
Exactly. In 2013, if I'm not mistaken, you kind of wrote a paper regarding kind of the short-sightedness of cancer research, in the sense that you kind of focused on the fact that it's very 2D, mostly done in petri dishes. What inspired you to think about it in that way?
I think it was more just seeing the shortcomings of the model systems that I was also using myself, that you think, wow, why am I culturing cells on a layer of plastic if our bodies are so much more than just that? And then you start looking in scientific literature, and you find that there is actually already a lot being done to make it better, but that it's not something that's at that time was very mainstream. It still needed to develop, I think, further, to also give others who are only working with the petri dishes and only the 2D layers, to also give them access to these model systems. So in that particular paper, we really wanted to show it can be done by anyone if you want it to.
Very good.
Maybe for the sake of clarification, could you tell what's the difference between a 2D and 3D cell model?
Of course. So if you take a 2D model, you basically have cells that grow in a single layer on a surface, typically plastic. Whereas if you go 3D, then it's already in the word sort of, you don't have one layer, you have multiple layers of cells, and that's is in a way a little bit more complicated to culture, but it also opens up a whole new world of biological relevance.
Very good. And yeah, maybe we could delve into the culturing specifically. How is that done? Because if I think about it, and of course, we learned with Petri dishes with, let's just say, generally when we observe things, we use a microscope and it has to go on a slide and it has to be very thin and everything. How would we culture this 3D thing? Because in my brain it's very hard to imagine how to do this kind of effectively.
Well, it depends a little bit on what material you are working with. So in my case, I work mainly with cancer cells, and that can be cell lines that have been around since, 1960s, 1970s. You can freeze these cells down, start them up again, and that's, for me and for I think a lot of other researchers as well, the starting point. Okay. It's reasonably simple, reasonably practical also to do. And those cells have been adapted to grow in a Petri dish or to grow in a flask like we have these days. We've gone a little bit more fancy than just a Petri dish. But the cells, they will stick to the plastic in a layer, and cancer cells, they have one purpose in life, and that's to proliferate and grow.
Exactly.
So in no time, you typically have your flask full. And then, you can put them in new flasks, more flasks. You end up throwing cells away even because you get so many that you just can't keep them all, so to speak. And then part of the cells that you culture, you use for your experiments.
Very good, very good. And so, let me think, where to go from here? Specifically now, I guess we can transition even already to the organ-on-a-chip model, because I feel this very nicely relates to how this idea kind of started, and maybe what you think about it to begin with, and how to explain organ-on-a-chip to just a layperson.
Yeah, so what I ran into, I already mentioned, we grow these cells in a flat layer on a plastic surface. Okay, that's maybe not that representative, so we go three-dimensional. Okay, we start to approach us humans maybe a little bit better. But then you also run into the thing that, I'm interested in drugs. I want to see what therapeutics do on the cells. Then how do I do that in my model system? Do I just take a drug and dump it on the cells and here you go, good luck, and we'll see you in 24 hours and we'll see what has happened? But that's not how we treat patients, right? Yeah. Patients get treatment in cycles. They get treatment and then they are taking off again. They get treatment again, and that can happen 6, 7, 8 times, or if not more. So how can we recapitulate that in our cell culture systems? And that's where my journey in the so-called microfluidic system world, so the tiny channel Petri dishes, so to speak, where that started, because those systems enable you to also have liquids flow over your three-dimensional model, and you can add drugs to that liquid, and you can choose when the drug is there and when it is not there. And in that sense, you can really nicely model the dynamics, so that the cyclic exposure that you would have in a patient. So that's where my journey into the sort of organ-on-a-chip field started, that I really had a problem with how we perform our experiments. How can we solve this? Well, maybe this is something worthwhile to try. That's for me where it started.
And I'm wondering, because you said that you would have the drugs administered to the organ or to the model, to the chip or 3D chip, but you said that it's not possible to administer this drug in cycles with the 2D or 3 models. But could you not maybe, I don't know, have some connection or tube to it that you would just administer it in that way? Is it that difficult, or what is the challenge there?
The challenge there was that when you grow, in our case, we grow small avascular, so without any blood vessels, but small tumor clumps, clumps of cells. and they are surrounded by the cell culture growth medium, so all the nutrients of the cells. But it's very difficult to replace that medium every single time, and you run the risk of damaging your cell clump. And trying that, it's just so impractical that you're welcome to come to our labs and have a go yourself, but it's really difficult to do. So we were looking for a system, how can we do this in a more sort of sophisticated, more practical way? And that's where a chip system, it's just, sort of as if it came falling from the sky like a godsend. This could help us really do what we want to do.
So what is the main difference between a chip system and a petri dish? Is it, I mean, for example, the way you described it, it seemed to me that a petri dish or any 2D or 3 model is just throwing the cells basically in the dish and hope for the best. But is it that organ on a chip is more we structure the cells in a way?
It's more controlled.
It's more controlled.
More controlled. And the lab where I work, we are a prototyping lab, so we have facilities to make and design chip systems ourselves. So it's really fit for purpose. So whatever biological question we have, we can translate that to a chip system that would fit to the research question best. And in the case of the drug exposure, it's different from the petri dishes that we use, different from the plates that we use, in that it is smaller, so we save more material. It's also differently configured with channels that you don't have in your well plate, with reservoirs that you don't have in your well plate. So it's really something that we have control of, what we make of it. And that's not really the case for the plates. Those we just buy. We still use them for certain purposes. But the chip systems are really fit for purpose.
And when you say a chip, I can't help but thinking in terms of computer chips, because I'm the computer generation, the 2000s. And so is it more like a computer chip? Is it a thin chip, or because I know that there's the way that it's made, it's called lithography or something, and then you fabricate it with lasers or something like that. I'm not sure. Maybe you could explain more about it. But what is the connection actually?
Well, actually, this field started off being inspired by computer chips. Also, the extreme miniaturization that we have seen in computer chips that now enable us to all have a mobile phone that fits into our pockets. That miniaturization is typically, actually the same what you could do with liquids running through channels. So that's what was done in the microfluidic field. So that you don't have a large stream of liquid, but you have narrow channels with the size of a human hair that you can send liquid through. And that miniaturization has also enabled us, as me as a biologist at least, to exploit that into our own field. So with chip, we sort of mean a small plate of plastic or any other material that we want to use, where channels have been made into, and we can use that to grow cells in. And for the fabrication, what you asked, it depends a little bit again on what question we want to answer, because we have, let's put it this way, depending on the size that we want and the material that we want it to be made of, we have different approaches. So one is to just take a slab of plastic, like plexiglass that we've seen in the supermarkets all over during COVID. And we can use micro milling, so a milling approach to take away any material that we don't want. So you basically dig out the structures in the plastic. And in that way we can make a chip system. But also, as you say, we can use the clean room approach with the lithography, where you sort of start from an initial design that you then transfer to what we call PDMS. That's a material that's elastomeric, so that has different mechanical properties than plastic. and you sort of end up with, I always call it a muffin tray, sort of a baking tray that you can pour your material, your PDMS in, and then you can peel off the structures that you want. So it's really making a master mold that you can then have your channels or that you use to create your channels. So it's a different approach, and it really depends on your research question which one you would use.
Because that also got me thinking that if we want to mimic the body and we want to mimic the medium in the body, we're putting now the cells on, as you said, plastic or any other medium. But I'm thinking, what is the medium that the cells in the body reside in? Because the basic unit is a cell, and they're residing within cells with cells, so it seems there is no medium in the body, or is it?
There is, but it also depends very much on which tissue you are trying to model. Bone is the hardest tissue that we know, so that's also the closest to plastic, even though our bones are still not as hard as plastic. But the brain, for example, is very soft, so therefore that would also need a different approach if you would want to model that in a lab. But all these cells, they are connected to one another via extracellular matrix, which is a biological component that provides it the structure, like the scaffolding around a building. But even that differs in composition between different tissues. So I think our body is just a fascinating piece of engineering if you look at it from that point of view, to make it function in the way it does.
I'm wondering, can't we just replace plastics or like put on the plastics extracellular matrix like collagen or proteoglycans and other things?
Yeah, you can, and that's also what we and others do in our labs. If we want to take a model system from 2D to 3D, you can use one of these extracellular matrix materials to make it three-dimensional. The only thing there is also, which ECM do you choose?
Exactly.
Again, it's fit for purpose, so you choose the ECM that fits your tissue that you want to model best.
And do we know the exact composition of each ECM?
Definitely not. In the body, no. Because
it also determines the softness and the stiffness of the human tissue.
Yeah, exactly. Yeah.
What would you say is the most difficult ECM to replicate in terms of the body?
I think there is not really one particular ECM that's most difficult. I think the difficulty is in that it's a mixture, it's a composition, it's dynamic, so it also changes over time, and that just makes it really difficult to grasp. But we start somewhere, and then you build up in complexity, and then
What cell actually, or what kind of cells are responsible for the production of ECMs?
That can be different cell types. Mainly it's the mesenchymal cells, so the fibroblast cells, so the supporting cells. And that's also what you see, for example, in diseases like fibrosis, that there's an overproduction, an overactivation of these cells, and that they produce excess matrix, and therefore, you also end up in trouble. Yeah.
And is it an idea maybe to just extract these cells and put them in the medium and then just let them do the work, even if we don't know what they are doing?
And then let them produce the ECM.
Exactly.
Yes, you can, yeah.
Is that possible, 100%?
Definitely. You can culture cells, take the cells away, and then what's left is also extracellular matrix.
And is it practiced in the labs? Okay.
Yeah.
Oh, that's cool. So it's well thought.
That's definitely, it's not science fiction.
Yeah. And relating to the lithography and the materials and the mediums that we have talked about, I have also came across something that's called the smart materials for organ and chip. Is that something that you have heard of?
Depends on in what context you mean smart materials.
I actually did not. It was just something that was mentioned along the different components that organ and chip could be manufactured of, and I did not quite understand it, but I found that was interesting. Smart material, anything that has smart into it. So I don't know. Is it something that's commonly referred to in the literature, or
not to my knowledge? But a smart material for me would mean that the material is also dynamic and that it responds to cues. But I'm not entirely sure in what context you've read it.
Yeah. Well, in that case,
we could
just move on. On that
Generally, the thing I think that most of us kind of see as the true future of organ on a chip is perhaps replacing certain techniques in drug discovery that we've been kind of, I guess, we are familiar with and we find purpose in, but there is obvious improvements that can be made. And generally speaking, I guess, sustainability is one of the main things that organ on a chip is trying to tackle. How so? How does this technology kind of create a more sustainable drug discovery?
Yeah, I think the drug discovery process is in itself quite inefficient. We have a lot of compounds that are tested, and what actually ends up being available for patients and successfully available for patients, it's meager. We're not doing a very good job there, I would say. That also makes it not very sustainable because it costs a lot of money for very little gain. I think if organ on a chip keeps developing the way that it does, it can be a tool that can help predict whether a certain compound might actually be successful in humans. And in that sense, it might save you a lot of time and effort putting compounds through the whole drug development process that may not even make it. So I think it's about making better predictions on both the efficacy and the safety front. On what will actually work in humans in the end.
Okay.
So as a sort of an intermediate step, I would see it.
Okay.
So if I understand it correctly, it is a sort of filter where we could then filter out the in vitro experiments and see what is fit and what is the most suitable to be carried out in vivo. And then we could focus maybe our effort and money into a smaller set of compounds.
Exactly. Yeah.
So basically, we're not replacing the animal models completely. We're just filtering and reducing the animals.
I find that always a little bit of a difficult question because animal models are not just there because we want them to be there, but it's also something that regulators typically ask. And that's because that has gotten we've gone to rely so much on that in rules and regulations that it's difficult, I think, to see organ on a chip as a total replacement for animal models. So that I would find difficult to predict. There are some how do I put it? I'll rephrase it. The FDA, so the Federal Drug Administration in the States, is making a move towards animal-free testing. In Europe, I'm expecting that to also come. But then it's still, what would you then use? That's the difficulty. That's the difficulty, right?
Yeah.
And I think at the moment, the organ on chip field is not yet there. I think there are still more case studies to be done that can really say, we've benchmarked our organ on a chip system against animals or preferably even humans, and we can make accurate predictions. I think we need more case studies that actually show that in order to convince everyone, including the regulators, that these systems are valuable and that we can use them also for accurate predictions.
But I think also I agree that replacing animal models 100% is not a very logical goal, because we do need to test it on something as an intermediate step, and we do eventually also test it on humans, clinical trials, and we're not going to replace even humans subjects because we need to test it on something. But maybe the severity of these tests could be reduced.
Could be reduced, and it's I think a complicated issue because what you've seen in the cosmetics industry, for example, is that animal testing was banned, right? It was not allowed anymore. But then, they were also struggling with what do we then, what will we then use in order to get new compounds on the market? And And yeah, that's a dilemma that I think we should be aware of and that we should think carefully about. Yeah.
Yeah, we can continue for that. What issues specifically can organ on a chip solve that, I guess, animal models have inherently from the very beginning? Because that's, I think, something obviously for regulators specifically that'll be a very big, I guess, question. So is there any like specific area within kind of animal modeling, general animal testing that organ on a chip is much better at solving?
I think a key advantage of organ on a chip compared to animal models is that we can use human materials as the input. So you start off already with a genetic background that is human. And although obviously we differ genetically not that much from mice, our appearances apparently show that small percentage that we do differ has an enormous impact. So I think moving away from animal origin, going to human origin, that's really also one of the things that gives organ on a chip, hopefully the power that we think it has.
Fingers crossed. Yeah.
Perfect. And now we have talked about organ on a chip a lot. Maybe you have also a model for organ on a chip.
I brought one, yes.
Yeah, it's probably bigger than the organ on a chip we use.
Yeah. Yes, this is not really a microfluidic model anymore, but more a macrofluidic model. But this is one that we made for visits to, for example, primary schools, because I think it's also really interesting to see how these kids respond to science and a scientist in the classroom, and I just love interacting with them, so I do that every once in a while. And then this is one that I bring. It's just a very simple Y-shaped chip with a Y.
Maybe even hold it up to the
Y-shaped channel.
Can we see it on the camera?
Perhaps.
Okay, great.
It's a Y-shaped chip, so we have two inlets on the left and on the right, and one outlet. And this is just for these kids to also already hold it, experience it, and see what is this now. And then what we did at one point is mix lemonade. So we have lemonade in one and water in the other, and then out we can fill cups with the drink. So that's just a really fun way to show, what it looks like on a macro scale. And obviously the real thing is a lot smaller, but at least with this one you can properly see what it looks like.
Already I guess like the pharmacokinetics of it can be shown quite nicely.
Exactly.
Which is very cool.
And so the key concept here is that we have these channels, right?
That you have channels.
So we can relate the different cells to each other. Like this is, for example, we have a neuron. This is the axonal part, and this is maybe the neuron itself, and so we can and this microglia maybe. So these structures could be defined well with this organ on a chip.
Yeah, define the structures, how you organize the cells, but also control of flow, like we have in our body, our circulation. That's something that we can also replicate with systems like this.
Fantastic.
Like the mechanical signals, you mean the air and the blood and
that's and blood flow exactly. So how that the stress that puts on cells you can replicate with this. So there's an awful lot of things that you can do with these systems. Yeah.
Is it usually more complex than 3 channels? How many channels could we put in there?
How many you like?
Really?
Yes.
So it's
how many you need?
Okay.
Yeah.
Wow. Great.
Yeah. No. Usually it's a little bit more complex. Obviously, you try to make it as simple as possible. Again, you try to make it fit for purpose, because there's no point in making 1,000 channels on a chip if you only need 2 of it, right? But usually they are a little bit more complex, also because it's on a smaller scale. So then that also makes it more difficult, for example, to connect the tubes. And I mean, our fingers are also a certain size, so you don't want it to become too finicky, right? It needs to stay practical in order to use it.
Perfect. I think it's a perfect time to take a break, and then we could go back to continue our conversation.
Thank you. Welcome back, everybody. We had a lovely break chatting about lots of things, but now we want to get back into organ on a chip, and specifically its applications in practice. How can we do that? And specifically maybe starting with something we talked about in the previous section, about, for example, tumors and tumor metastases, for example? How would we look at that?
Well, yeah, that depends on what your aim is, what you want to study. So what we are doing in our labs is that we look at this repeated drug exposure and what effects that has on the cancer cells, and whether by treating it in that way we might actually trigger resistance. So we're looking at mechanisms of resistance. So it's still quite, I would say, fundamental biological research that at least we are doing with our chip systems. So it's what you want to do with it. That's, I think, the main question. So in our hands, we really look at mechanisms. So mechanisms that can cause certain malignant behavior in the cancer cells.
Very good. And that's more basic science.
More basic,
but
yeah.
Going more to the clinical area, is it possible to use it more to apply it for example, a personalized medicine? I don't know, and maybe I'm fantasizing now, but for example, then you take a chip and then you take stem cell, you put it on the chip and you see if the cancer will metastasize, or you see if the drug will, or the immunotherapy, for example, will work on the cancer. Is that feasible at this point, or is it not?
Well, for sure you could take a sample, for example, someone that has cancer, and you take a biopsy and that you culture that biopsy in a chip system. That's for sure possible. The only thing, the hiccup that I see there is the timing. Like, for example, if you want to show which drug works best, okay, have a patient, we take a biopsy, we put it in a chip, let's say it's successful. We need to expand it because we want to test multiple drugs. We need to test the drugs. But ideally, I want that patient to already be treated immediately. Yeah. Okay. So I see a few practical limitations with that approach. But that being said, why not look in our chip systems in parallel to patients, for example, being included in a study? We trial that approach and we look at biomarkers. Right? We could see in our chip systems, can we find biomarkers that signify whether a treatment will work or not? And maybe in the future we can then correlate that to potential biomarkers in patients. And then we know from our experience, okay, that patient would fit based on a biomarker profile in that particular cohort, and we need to treat it like that.
Okay, so we use it for studying the biomarkers and the relation of biomarkers to the efficacy of the drug.
Yeah.
And then instead of doing the whole organ on a chip with the patient, we just look at the biomarker.
Exactly, yeah, so that you have an intermediate step, but that you save probably a lot of time and you can immediately start treating the patient. You could still combine it with culturing actual tissue from that patient on a chip, but then at least you can already make a start. And you don't have to wait for results to come in.
Yeah. So that's just generally for streamlining the process of immediately treating someone, is kind of, I guess, applying this brute force method of, okay, let's test all these markers related to this disease and at least have a nice kind of preview or potential look into the future of, okay, this might work initially, and then perhaps take the stem cells. Okay, that's very interesting.
Yeah, exactly. And I was thinking maybe also different genotypes can express different biomarkers, and then we have the capacity to discover all these genotypes, right?
Exactly. Yeah.
Oh, okay. And relating actually to that, to the biomarkers thing, is it the case that when we have organs on a chip and this 3D structure and the structures we're putting it in, that they could, for example, produce different factors and biomarkers than 2D and 3D cell models?
Oh, yes.
Yes.
Yeah, and also 2D versus 3D. The biomarker profile will also completely change.
What I can't think what is the reason for that. It's the same biological components and same...
Yeah, but the environment, that's why the environment is so important, right? The environment changes how you and I even behave, and all that. The same goes for ourselves. So if we put them in a different environment, and then 2D is a different environment from 3D, and 3D versus structured 3D is also different, then the cell behavior will be different, and therefore also, whichever biomarkers they may produce. or they may have. Yeah.
But in that case, I'm wondering now, isn't it better to have slices? Because then we have the whole 3D cell layer in a structured way as it is in the body. Why do we need the organ on chip in that case?
It's a different approach because with the slice, I can't play around with it that much. It's there, it's complete, but I can't change, for example, if I want to change ratio between two cell types that are present in the slice, I couldn't do that. I can do that in my organ-on-chip system by just mixing them in a different way. But the slice, it's a very valuable model, that be said. Otherwise, I'm in trouble with my colleagues. It's a very valuable model, but there are certain things that it can't do. And the same goes for organ-on-chip. There are certain things in the slice that an organ-on-chip will not be able to reproduce. So I think both have advantages and disadvantages and limitations in that sense.
No, but regarding complexity, I'm imagining that slices has the advantage over organ-on-chip.
Yes, yes. Unless we are able to fully mimic all the cell types that are present in that slice in the same configuration, in the same structure. But that's still,
How close are we? Or can we know even that we are close or not?
Well, I think in that sense, we also need to look at how well our models perform. And then slices could be a very interesting one to benchmark it against. And to see, how well are we actually doing? But I think, in certain fields it's getting there, and in other fields we'll probably need a few more years in order to figure it all out.
Because we also we were having this discussion, I guess, yesterday, that, for example, you can model a specific organ, but some things in the body, for example, the immune system, are distributed all over the body. They're not in one organ. So how could you capture that complexity in organ-on-chip, or how can you add that element, for example, of immunity and immune systems, immune cells?
Yeah, I think immune cells, it's so complex, right? You have so many different types and so many different ratios. And indeed, as you say, it's again a very dynamic system, and it's all over the place. It's not like it's an organ that's in a certain in a certain place in your body, and that makes it difficult. But then on the other hand, the advantage that organ-on-a-chip systems offer is the flow again. So we can actually flow cells also through the system.
Smart.
And not that we are there yet, that we can capture even only a fraction of the complexity of the immune system, but it does, I think, offer a huge opportunity to look at interactions between resident cells, so cells that are sitting there, and the cells that are actually circulating.
So that's still, I guess, to be determined, or a to be determined part of the research, because, I assume it's an endless rabbit hole. You could write just thousands and thousands and thousands of PhD theses on that kind of point. Specifically, another kind of limitation that we came up with when we were discussing, aging tissue and senescent cells. Can we somehow put those into the model? Because, of course, many of the people, especially with cancer, that we're going to be treating are older patients. And so we kind of, I guess, also need to try to model the environment they find themselves in, or the kind of cellular environment in their body. So can we already do that somewhat reliably, or is that also quite tricky?
And then you mean in the way that we can we recreate an environment in a chip system?
Yeah, so for example, if a cell senescent cells, I guess, cells that kind of have been around for a long time, can we somehow recreate those? So from my minor genetics knowledge, cells that, for example, have shorter telomeres already, genetically speaking, can we put those in, or is that also kind of still
or maybe
quite
just
tricky?
modeling the aging tissue? Because what we have, I believe what we have also, if we have it from stem cells, is the younger phenotype.
Yeah.
And I don't know how we could mimic the aging phenotype.
Yeah, so from stem cells, that's, I think, still a challenge.
Yeah.
Because differentiating stem cells, as far as I'm aware of at least, still creates phenotypes that are a little bit, not fully mature. Well, maybe we can still optimize that with protocols. But to really mimic aging tissues, I would say, then we can also start with aging materials that we could then use to put in a chip system. But I think the key, well, the fascinating stuff, what are the processes that actually trigger aging? And that's something that you could, of course, study also with chip systems. That you start off from not yet aged material, and that you see which triggers provide the cues for this tissue to actually progress towards, for example, like you mentioned, senescence. So can we identify then the cues? And you can use it again more as a mechanistic model system to see whether it's possible to, or which cues are needed in order to progress. Yeah.
And so are we at the point where we could, where we know how, for example, to speed up the aging of a tissue when we have, for example, a young phenotype, we know what factors to put in so that we could measure the aging and stuff? I don't know.
Well, we can, well, senescence is an area that I have not worked in extensively, but we can induce it. Like we can take cells and induce senescence. But from what I know, that's still quite artificial. So you really aim, okay, senescence is my endpoint, and it doesn't really matter how I get there. So you can use, for example, cancer drugs even to induce senescence, but that's obviously, hopefully, not really how
it works.
Not how it would work in our body. Yeah.
Yeah. Wow. Okay, so that's also another. Yeah, I guess when we were thinking, we assumed, okay, this is horribly tricky, and I don't know how we're going to reach it, but with extra work and with how you've broken it down, I guess it is a possibility for the future.
Exactly.
That's quite curious.
Yeah, yeah. And also, well, I know that organ chip are becoming more complex, and I was wondering maybe could we do vascularization? Can we have like nurture the blood, the blood vessels and connect them in a way to the other organs that we have the actual flow?
So you would say, oh, I want to make a system with two organs that are connected with a blood vessel. Is it?
Yeah, because I think that's more realistic, right?
Yeah, that would be definitely more realistic. So blood vessels on a chip, they exist, and they're functional, and they can also be used for perfusion. So it's a matter of coupling different organs together. Which is not trivial, I should say. But from a practical point of view, it could be done if you have one system for one organ, another system for another organ, and you connect it with a blood vessel. That could be done. Yeah.
And then do we need to? I don't know, but like whenever we have the blood vessels, I assume that we need the heart as well. Do we need the heart on a chip always to be present when we doing the vascularization, or no?
Well, like a pumping heart, you mean?
Kinda.
No, for our chip systems, we usually use artificial pumps that, do the perfusion of the whole system. I'm also really wondering, like, if you would want to have a beating heart on your chip. I think that would be really, really complex. I'm not entirely sure whether anyone has ever tried. But I could imagine that you run into issues also with, you need a very thick layer of cells in order to get the pumping.
Pacemakers, yeah, exactly.
To get that done. I'm not sure whether that has been tried. It would be really cool, wouldn't it?
I think so, yeah.
Yeah.
And I was maybe wondering, we could also have two chips, and then we could couple them together. One of them is a model for the heart, and the other is a model for anything else, really, we would like to discover.
Yeah. Yeah, that would be really cool.
Okay.
Yeah.
So I think there are still a lot of ideas to be discovered.
Too many ideas.
Too many ideas.
Yeah.
This is just an endless topic. You open up the can of the worms, just all come out, and it just never ends. Wow, fantastic. Yeah, so I guess, do we want to get into a bit of a interesting, I guess, philosophical question regarding specifically the brain?
We could. But I also still had one question, because we're still touching on the area of limitations and trying to see what we could do and what we could achieve. I was wondering, all the measurement systems that we have right now are designed for basically the Petri dish.
Yeah.
So I was wondering, because I worked one time with the flow cytometry, and you put the Petri dish in, and it analyzes basically the cells and what is in there, and it shows that on the computer and does it very nicely. But I don't know if you could do the same with the organ-on-chip model, because these systems has not been implemented really, or even produced for organ-on-chip.
Yeah.
So it depends very much on what you are trying to analyze, what technology you would want to use to analyze your system. There are a few issues that you may run
As
into.
always.
That is the miniaturization. So miniaturization is a key aspect of these systems because it saves, if you have scarce patient material, it saves you material. You can do way more analyses. You would think of that as an advantage, but on the other hand, for your analyses, it's sometimes a huge issue because you are just below limits of detection because the sheer amount of material that you get out of your chip system is not sufficient to reach a certain threshold. There are techniques that you can use, like microscopy, for example. A lot of chip systems are suitable for microscopy, and that's really funky because you can see in real time what is happening to your cells, and if you expose them to a drug or whatever, then you see them crumbling and you see what happens. But yeah, it's again a bit of a question of what technique do you need, and yeah, which fits your purpose best. So microscopy might be good, but it might also be an issue because your chip is just too thick, or the tissue that you make is too thick in order to see anything. But techniques like ELISAs might not give you the limit of detection because your material is too scarce. But then I'm a half or a glass half full kind of person. There are also a lot of efforts being put into developing new techniques that are more suited also for chip systems so that you can get the information out that you need. And if I may give an example of our own practice, we've run into issues where our miniature tumor clump that we are culturing becomes very difficult to do microscopy on because it's, a little bit too big, a little bit too dense, and the microscope, the light is just not penetrating properly. But then we thought, what are we actually interested in seeing? Well, that's sort of the middle layer of our little tumor chunk because that's where the interesting parts happen. Well, then let's see if we can build another chip where you only culture the middle part, right? Where you give the cells a chamber, a reservoir that they can grow in that's only resembling what we want to see, and that is suitable for microscopy. So it's also, right, a way of thinking what could we do to make it possible what we want to get out of, and to adapt your system to the technology. And on the other hand, the technologies are being adapted to the chip system. So it's sort of a both worlds that we could make use of.
So what is a measurement system or an experiment that you would think, oh, I would just do it with in vitro 2D, 3D models that are already existing, that are already going for organ-on-chip?
So you mean in what situation would I not use a chip, but would I use a well plate? I would go for a chip where dynamic conditions are needed, where you need the flow, where you need, where it's not possible to use a well plate. But like, for example, the Petri dish, the well plates, we also still use to just grow the cells, to have enough cells to put in the chip system. So we also still use them in that respect very, very often. And it's still, a good technology to use. But then when we do that, we also take it one step further and then do the chip experiments as well. So it's really, is dynamics, is flow, is that necessary to answer your research question? If that's a yes, then I would go for a chip system. If that's, well, maybe I don't need it, then a well plate may just as well suffice.
So there are still advantages to the other systems. We're not getting rid of all the other in vitro systems.
No. And I would also not argue that organ-on-chip is the holy grail that everyone should use. No, definitely not, because it's a lot of effort that you put into making a system function and into getting the data out. And if in your case a well plate works just as well, then why not?
Perfect. Interesting.
So just, one can also look at it as not something that's going to replace everything, but just another tool in our arsenal that's now just filling up massively with so many interesting new developments to kind of detect these things and to not only diagnostically kind of look at things, but also prognostically. It's very fascinating. We find it really interesting. So do we want to delve into philosophy now? Or?
Let's go. Let's go.
So, of course, we did explore, I guess, the fact that, we can put a whole organ or, the environment, but then, I guess, hypothetically, if we could put an organ on the chip, and if it could become advanced, then obviously ethical issues arise from, for example, a brain on a chip. With the advancements that could occur in the future, I guess, would there be ethical dilemmas, I guess, for yourself, maybe, just to get your opinion first?
Mm-hmm.
And then, if we could prove that this was a truly functioning brain, so aside from any AI anymore, this is like, okay, this is actually neurons that are firing, true neurons, not just, silicon chips that act like neurons, true neurons firing and creating thought by themselves. Do we then kind of start, I guess, creeping into territory where we really shouldn't, I think, but maybe you have a different opinion.
Yeah, I think before we're there to actually get a fully autonomous brain, I'm not sure if we will ever get there, because it's so immensely complex. And if that would be the case, let's say hypothetically, we are able to make a brain that can think for itself. I'm also wondering then, okay, then that brain is sitting in our cell lab incubator, and then, where will the information go? That's I think, right, it can't really go anywhere. So I personally don't really see huge ethical issues unless that brain starts taking over like some sci-fi movie, taking over the entire lab for, doing bad things. But I think as long as it's just there as an autonomous brain, I see it as a fascinating tool for research on which we could learn a lot about neurological problems. So I personally, I'm not too concerned with that ever becoming a problem. And if we would have it, then I think we should make use of it.
Maybe if we're going more into the direction of placenta and embryo and stuff on a chip, maybe this is where we could run into ethical. As I know, maybe placenta on a chip, is that possible at this point?
Rudimentary placenta, yes. So you can culture placental cells in a chip system and have sort of a barrier where you have the placental cells on one side, so you make a maternal compartment. And then endothelial cells on the other side, so you make a fetal compartment, and then you can see, for example, again, drugs, how many cross, how many stay in the maternal compartment, how many go to the fetal compartment, how many may even accumulate in the placental cells. But again, placenta is also quite a complex tissue that I think still needs more work in order for it to be recreated. And the added challenge there is that it changes, right? The placenta initially in the first trimester, for example, is very different from one in the third trimester. And to also recreate that on a chip, I think that's, I find it exciting, but still a really big challenge in order to accomplish it.
So it's really dynamic.
Yeah, it's very
dynamic.
Very dynamic.
And do we know all the factors that we need to put in so that we could model the placenta and model the growth of the placenta?
Very rudimentary, I think, but not yet to the extent that it fully replicates, what happens in humans.
And so basically, the barrier for placenta is the growth of placenta that we could not really model.
I think so. Yeah. That it's so difficult to grasp what components are really needed and what triggers the placental cells to do what they do, to replicate that in the lab. It's challenging,
Even with the other models? For example, say we have an ex vivo or something.
But then still ex vivo you get from a person, right? So that's already at a certain stage. So then you would need material from all stages to see what happens and to be able to put a mechanistic idea behind what is happening, to know mechanistically what are the cues that we need to put into our chip system in order to accomplish what happens in vivo. That's very complicated.
Because that means that maybe if we do an experiment on a placenta at a certain stage, and we find out, for example, that this drug does not cross the placenta, the barrier, but it could really be at a different stage. The placenta has a different composition or structure, and then the drug starts to be toxic.
Yeah, that can happen. Yeah.
And in that case, I don't know. In that case, maybe it's just better to have in silico models. These are the most dynamic that I can think of with the limited knowledge that we have, right?
Yeah, but then also for in silico models, you still need input. So you would still need to know either from patient data or from in vivo experiments or even from in vitro experiments. You would need information to put into your model, right, in order to make assumptions that are relevant.
Yeah, so it's kind of a paradox, actually, because we need to expand our knowledge, but in order to expand our knowledge, we need knowledge.
Yes.
It's basically model in silico.
Yeah.
Endless paths to go down. Wow. Yeah, I wouldn't know where to start personally. So it's fascinating that we've even gone to this point. And like, generally speaking, what is the current price of production of an organ on a chip? I know these are kind of like, at least here in Groningen, we're working on kind of prototypes and we're testing those. But yeah, I guess, what is the current rough kind of estimate on how much it kind of costs to create one for kind of functional use in, let's just say limited to maximum of, I don't know, 5 channels or something like that. But I assume more.
It depends on the design, on the material.
Again.
And it also depends, are you making it yourself or are you buying it commercially? Commercial chips can be quite expensive.
Yeah.
There's also cheaper ones, but I think there are out there that cost 500 euros per chip. Obviously, if you have a prototyping lab, you can do it a little bit cheaper, but that does not take into account the cost that you have already spent on machines, on people, right? So it's I think difficult to put a price on in terms of the materials that we use. It's not too bad, but all the costs that are not taken into account in the material, that I think it adds up still quite hefty.
I can imagine. Still, super tricky question to answer. And I guess the kind of forgot to highlight this, but the whole idea of the organ on a chip, and you described this very kind of beautifully in a piece you wrote. The kind of interplay and interconnected kind of nature of this project, and I'll kind of throw a quote you used back at you. Usually you do not ask a plumber to install electricity or an electrician to paint the walls and window frames. However, bringing together the expertise and knowledge of different professionals results in a house in top condition. And the same holds for our idea here. I guess maybe stress the importance of perhaps even on a weekly basis, how many people you interact with and from different fields, and how much kind of this, I guess, interplay is important for developing this, because I can assume it's the need of different expertise is endless in this regard.
Yes, it is, and especially in the early stages to set up chip systems, right? You need people, engineering people, that know how, and it can be even very basic, like fluid flow. Right, that you need to be able to model that and to understand. Okay, I put fluid in these tiny channels, but what actually happens? It goes from there up to, I would say, a clinician that has a clinical problem. So, well, okay, I need to solve this, and maybe it could be done with a chip system. And then people in between, like biologists, like myself, but also chemists, right, because we want to do analyses. So it's a whole range of people that if you, I think, want to have a successful chip system, that have a place in this field. But it's so important that they keep communicating, because we speak different languages, I have the feeling sometimes, right, that the clinician doesn't really understand the engineer, right, and a biologist can't really mediate between that. So I think it's all about communicating and understanding what are the limitations or what are the challenges and what are things that we can already do, and how can we develop it further. And I think that that's really key. Keep communicating and seeing where do we have mutual interests and mutual grounds, and where can we help each other further. Right? With a clinician coming with a clinical problem, and an engineer thinking, yeah, oh, maybe I could come up with a solution for this. And then a biologist seeing if they can integrate the biological components in there.
Yeah, because I know that you, we talked about that, that you like the multidisciplinary approach.
Yes.
Yeah. But I'm wondering, at the research level and at the research units we have currently, do we have good multidisciplinary structures, functional structures that we could, that are now present?
Yeah, I would say so. We have theme groups, also broader in the Netherlands, broader even in Europe. Theme groups that work on organ on a chip, so it's also people that have mutual interests, but coming from different perspectives. So people more from the manufacturing, the fabrication, the design, that I'm just saying maybe focus on new materials, right? That could be of interest. But then also from more the clinical side, where there are really issues, research questions or even clinical questions that maybe could be answered with this. So it's really an interplay. But infrastructure, I would say, in the Netherlands, we are doing very well. Yeah.
And I was also thinking in terms more also of the maybe the literature. That should be understandable and accessible to all disciplines. And so, for example, the scientific journals are usually more specialized in one field. For example, it's like analytical chemistry or biology. Do we have maybe a journal that's just for organ chip that a clinician can go to, a biologist can publish in, a chemist? Do we have that kind of...
Oh, you'll be happy to hear that there are multiple.
Okay.
Yes, yeah. So there are journals specializing in lab on chip, organ on chip, that are really specialized, focusing on this area in particular.
And that's regardless of the discipline.
Regardless of the discipline, regardless of the application, it can be very fundamental. We made a chip out of a new material, or we integrated some kind of biosensor, but it can also be an application. Oh, we cultured patient material on this, and we see this and this therapeutic response, or more basic science. Yeah, it's possible.
Interesting.
Yeah,
I think that's that's a very good step, that we have these what do you call that, the shared knowledge medium that we could all access and we could all contribute to. Yeah. Oh, interesting. I think it would be a good time now also to take another break, 5 minutes break, and then we could go back to it. Now that we have talked about organ on chip, we want to move on to another related topic, which is extracellular vesicles. Doctor, what are extracellular vesicles?
they are small membrane-bound, particles that we think are secreted by every single cell in the body.
And what is the application of these vesicles that we have right now?
Well, the cells secrete them, so they contain a lot of cellular material, lipids, proteins, RNAs, some may even think that there's DNA in there. And they seem to have a purpose for everything. They're even put in skin creams and that kind of thing.
Okay.
I'm not sure if that application is actually working, but they are thought to have diagnostic potential because they reflect the cell that they came from.
Oh, the composition of the cell.
The composition.
Okay, interesting.
Yeah. And secondly, also potential as a drug delivery system because it's sort of a more fancy version of a liposome, if you like.
Yeah.
And thirdly, as they are packages of cellular material, thought that they may also have capacity to be therapeutics in themselves. So not with externally loading drugs into them, but as innate sort of therapeutics.
So obviously there is a wider range of these extracellular vesicles. But Mike, I was also wondering, how do we get these vesicles in the first place?
By culturing cells, yeah.
Okay, so it's not
at
difficult.
least if you want to get them for research purposes or laboratory purposes, typically you would start from a cell culture, and then the cells, secrete them in the medium, and you can isolate them from there. Alternatively, in blood plasma, for example, you also find a lot of these particulates.
And then going to the industrial, at the industrial level, do we have anything that could scale up this process?
Huge bioreactors
Okay.
of cultured cells, yeah.
And do we use it anywhere for therapeutics at this point, or is it just a promising tool that we hope it works?
Well, there are already some clinical trials that are evaluating it. It's just because they are so complex, it also makes it different from, for example, a single molecule as a drug, right? Because it's a complex mixture. And that makes it also quite challenging because you need a stable composition. Right? You also have quality checks with small molecule drugs that it's the same composition each and every time. And that I think with EVs is still quite challenging because it's a cellular product. So you need to make sure that your source, in this case the cultured cells, are also stable over time. That's still a little bit, still tricky.
But maybe is it an idea that since it comes from the body, from the cells, that we integrate it more with the personalized medicine approach, and then, I don't know, maybe we get it from the person or from the patient, and then we use it for transportation, drug delivery, or even, as you have said, therapeutics. Is that a feasible, practical idea, or is it only theoretically possible?
Well, I think that could definitely be done. Challenging it would be because, you need to sort out of this protein pool that is our blood plasma. You would need to sort out those vesicles that have the therapeutic potential, and you would need some kind of marker in order to do that, and that I think will still be quite a challenge. And these vesicles are only also a very minor part of all the material that we have in our blood. So I see a few challenges that we need to overcome in order for that to be a reality.
I think so. I think also the time it will take, because we need to extract them and then produce something from them and then give them back to the patient.
Yeah.
Probably this process is going to take months or years. And as you have said before, we need to treat the patient first.
Yeah.
Interesting. And does it have any applications in oncology? Because as you know, any new technology that we come up with, the first thing that at least I think of is like tumors, oncology. Anything can help.
I think as a biomarker, we talked about that before already. But even in our chip systems, we use EVs as biomarkers to see what's happening. But definitely in oncology, you can probably imagine that if you have a tumor of a certain composition and one of a different composition, that the EVs that they will secrete will also be of a different composition. The challenge there is, how do you identify which of these vesicles is of cancerous origin and which comes from another region of the body? That's still quite challenging. The same way that cancer diagnostics is also quite challenging.
Is it because that they do not differ much, or is it that we do not have the measurement systems, the right measurement systems to tell the difference between both?
I think it's a matter of both, although there are approaches that, at least in a research setting, are very well able to distinguish cancers from non-cancerous. But that is when you already have them separated. Right, then you can see the differences, but to do that in a setting where everything is pulled together, right, and not just normal EVs and not just cancerous EVs, but a lot of other lipid particles, proteins, cells, immune cells, everything together in order to then pinpoint that's a cancerous EV. that requires, I think, some technological advancement. Yeah.
But do you see a promising future for this technology?
Oh, yes.
Okay.
Yes, I think for diagnostic purposes, EVs are definitely an interesting angle to keep pursuing, and to also keep in mind of technological advances that are being made, like more sensitive types of analyses that could then potentially in the future be applied to the field of EVs.
Yeah. And what is the current research state of these EVs right now?
In terms of diagnostic,
diagnostic
purposes, well, it's not yet clinical practice. It's still in the preclinical phase. Obviously, the advantage of doing this type of work is that taking a blood sample from a cancer patient is typically not an issue. They have to donate so many samples already that an extra one is usually okay. But there's a wealth of information in there that can be exploited. So it's not that you need to burden the patient with an extra biopsy, with an extra operation in order for the research to be done. It's a matter of drawing a small sample of blood, one extra tube that you can then do the work with. So it's definitely an area that is evolving, but for it to become clinical practice, more is needed. Yeah.
So we still need some time, I guess, to reach that point.
Yeah, exactly. Because also the technologies that we in the lab use to look at these EVs, it's typically not something that you would find in a clinically diagnostic department, right? So also the methodology needs to be then developed in such a way that a diagnostic technician can just take a sample of blood and analyze it, and not just one, but probably from a whole battery of patients. So also in terms of infrastructure, there's still work to be done in order for it to become mainstream in clinical diagnostics.
Great. I think now we have already touched the future a little bit, and so moving on to what do you think is, or what do you want to pursue in the future? We have talked about organ on chip, and we have talked about extracellular vesicles. So what is the thing that you want to pursue in the future?
It's for me definitely pursuing the model systems further, thereby also using EVs as potential biomarkers to be also able to see whether there is a potential in that area, but increasing the complexity of our model systems. So we in our labs started off with developing the system, right? Developing sort of, if you like, the casing of what our cells will be in. Well, now we've put the cells in, we've put the drugs in, but from a biological perspective, our model is still quite simple. It's a lump of tumor cells, right? But you would want to maybe mix in other cell types, the immune cells that you mentioned, the combination with endothelial cells, so with the blood vessels, maybe even trying chunks of patient material to culture that in our chip system. So too much work, I think. A lot of work to be pursued in the future, but really to improve the biological complexity. That's really my goal for the next few years.
I think also for students like me and Oscar, it's always interesting to hear what are the challenges that are out there, so that we could maybe be a bit encouraged and motivated to know where to pursue our careers. As I always like to ask that question, what is the $1 million question that we need to answer in this field? Where is the big puzzle in this field?
Yeah, I think for me that would be therapy resistance. That's an area in oncology that I think is the key bottleneck. If you look at the number of drugs that we have at our disposal, it's like huge that you think, why can't we cure this disease better? But that's because you're trying to hit a running target. Yeah. So cancer continuously changes, and we throw another therapeutic at it, and the cells change. For breast cancer, for example, it can be that a patient has no symptoms whatsoever for 20 years, right? So you think, oh, the cancer is gone, and then all of a sudden, oh, a metastasis. So apparently there were still cells left that we had not been able to treat. But then once the metastasis is there, okay, but then how can we now treat this? Because it's become resistant to most of the therapeutics that we have at our disposal. So one area, and I think organ-on-a-chip systems play a huge role in their therapy resistance. What are the mechanisms and how can we prevent it from happening? Because that's I think, yeah, the part that we can't really still quite grasp. And if we can, then I think we could find a cure for cancer.
Would you think that these organ-on-chips in the future would have more promising potential within the basic sciences and, as you have said, explaining their mechanisms, or within the clinical more practices that you could use, as maybe doctors could use and lab technicians could use for analysis?
I think both. I think it's now still very much a research tool, but what we do see is that it's becoming more and more mainstream. More and more labs are using it, so we can advance the field in that respect as well. And then I think clinical application is going to be the next step. Yeah.
Great. Do you want to add anything on mind?
I think that's very comprehensive, and yeah, I don't know if I described this on camera, but just the can of worms that this is endless. It'll just keep expanding, and it's so fascinating. So I hope we, in the next few decades, we just keep moving forward with this, because it seems really promising and could really lead to some new ways of really kind of looking at reality, really kind of and seeing how to test things specifically, and more specifically. So thank you so much, Professor, or Doctor, Assistant Professor. Always the joke on this podcast. And it's been a lovely conversation, and we're so happy that you came to speak to us. And
yeah, thank you very much.
Thank you for having me.